Welcome to Biology Explained with me, your host, Mr. Hill, and welcome to our first episode of Season 2. It has been a while, so I do apologise massively for leaving you in the lurch after finishing our first unit. But I hope over the next few weeks we'll be able to cover all of Unit 2 of our National 5 biology course here in Scotland. Now, if you are in need of any support with your biology revision for National 5, or indeed for sort of GCSE or kind of any kind of sort of age sort of 14 to 16, wherever you are in the world, then please do hit the like and subscribe button so you don't miss these episodes. It helps me out massively, and of course it will help you out massively for your biology exams in the future. So in this episode we are going to be covering key area 1 of the National 5 Unit 2 course here, which is all about multicellular organisms and specifically about cell division. So in this video we're going to be looking at how the process of mitosis occurs, the use of stem cells, as well as the hierarchy of organisation in a multicellular organism. Okay, so without further ado, we'll make a start with the video. Please do pause, rewind, replay, make notes as you go, and don't forget to have a go at the question at the end of this video. Okay, best of luck everybody. We'll see you very soon. Okay, so let's delve straight into our first part of this key area, looking at cell division, and it's going to be the most important part of this topic, and that is, of course, the process of mitosis. Now, mitosis is needed for multicellular organisms in order to grow and repair new tissue, okay? Which kind of makes sense, okay? So if you cut yourself, or if you're growing, you're going to need more tissue to be made, and so your cells are going to have to divide to produce that. Now, mitosis is a very simple process. There are a number of different steps that you need to be able to put in the correct order and to use the correct key terms. So a bit of an exam technique here. This is does lend itself quite well to extended response questions or sequencing questions. So it's really important that you get each step explicitly said. You know, if it's a 3 or 4 mark question, then you've got to have to put at least 3 or 4 points, right? So it's important that you are able to sequence these different stages in this process of mitosis, as well as use the right words. Now, before we make a start, let us just remind ourselves as to why mitosis is needed. Okay, so I'm going to pop in this little box here, the reasons as to why we need mitosis. And it's quite simple. A multicellular organism, such as you, me, your pet dog, the plants on the windowsill, any organism that has more than one cell, needs to be able to repair itself when it gets damaged, and it needs to grow as it gets older. Okay, so mitosis is needed for the growth and repair of new tissue. Nice and simple, okay? You usually have to say both of them for a mark, okay? So just be aware of that, for growth and repair of new tissue. Right, on the screen we can see I have got some empty cells ready for us to go. So let's make a start on looking at the different steps involved in mitosis. Remember, the outcome of mitosis is 2 identical daughter cells. They are identical to each other, but they're also identical to the first cell here. Okay, so essentially it's like a clone, right? Okay, so the first step then in mitosis is that you will see that the nucleus actually sort of fades away. Okay, so we have our nucleus where all our DNA is found. The nucleus itself is going to fade away as the chromosomes inside start to condense. Okay, so remember your DNA is organized onto chromosomes, and that's where all your genes are found. And in humans, there are 23 pairs of chromosomes in each normal body cell. So the chromosomes themselves will start to become visible. That's what we mean when we say they start to condense. And you'll see them as crosses in this case. Okay, that's because they have duplicated before mitosis. The chromosomes themselves have duplicated. So the nucleus has disappeared, and I'm just going to just do a dotted line around that, just so you can see that the nucleus has gone, and our chromosomes condense. Okay. Next, the chromosomes themselves are going to start to migrate, and they're going to migrate to what we call the equator of the cell. So imagine the cell was like a globe, okay, and the middle line of our globe, of the planet, is the equator. And what we'll see is our chromosomes will migrate to there, and they'll line up along it, okay? So if I put my 2 chromosomes here, I've got my blue one here, and my red one, you will see that they will move to this line, this imaginary line, the equator. Now, using the same analogy, okay, as the globe, we start to see proteins grow from the poles, okay, and these are called spindle fibers. I'm going to start making them grow here. And the spindle fibers are very important in our next step because they're going to help pull apart our chromosomes in order for our two cells to have the same DNA. Okay, so stage 2 then, the chromosomes are going to move along the middle line, the equator, and the spindle fibers are going to start to grow from the poles. Okay, in the third step, what we're going to see is that the spindle fibers will attach to the chromosomes, okay, in the center bit where those two kind of lines meet in the middle of the cross. We call that the centromere. And the spindle fibers are going to pull the chromosomes apart. Okay, so in this diagram, they've already attached, and the spindle fibers are then dragging the different legs of the chromosome back to the poles. Okay, so let me just draw an arrow just to show that that's going on. So the chromosomes have been pulled apart, and they're being pulled to the poles like so. Okay, the spindle fibers have attached to the centromere. The centromere is just here. I'll put a little purple dot there. Okay, and they've pulled the separate chromosomes apart. Okay, remember, before mitosis started, your chromosomes actually duplicated. That's why they're crosses. Okay, so there's two copies there, and so they get pulled apart, and the separate copies are going to move to the poles of your cell. The last step, you'll have now your chromosomes in separate areas of the cell, and so the cell is going to start to kind of close itself. Okay, so the nucleus will reappear. Okay, so I'm just going to do a dotted line to show the nucleus there. The nucleus will reappear. The chromosomes will start to decondense. Okay, so they'll start to disappear, but I'm just going to leave them here just so you can see that they're there. Okay, and then the cell membrane starts to pinch itself until it's pinched completely, and then you've got two brand new cells. Okay, and that's the process of mitosis. So just to recap then, some key words that we're going to be using. In the first stage, we're going to say that the chromosomes decondense. They condense. Okay, let's make sure that gets corrected. They condense. Okay, in order for you to actually see them. Okay, and the nucleus disappears. The chromosomes in the second step are going to migrate to the equator. Okay, and we're also going to see the spindle fibers. Okay, grow from the poles. Okay, in our third step, the spindle fibers, I'm just going to do SS for short, okay, will pull the chromosomes apart. Okay, and in the fourth step, okay, the nucleus reappears. Okay, and, and the cell divides. Okay, and there you go. You've got yourself your two daughter cells that are identical to each other and identical to the parent cell. Okay, don't forget those words: condense, decondense, spindle fibers, equator, and poles. Okay, so if you want to redo that, by all means, pause and go back to the start of this section. But we're now going to look at, just to focus on a little bit about the structure of that chromosome, just so we're clear about what a chromosome and a centromere are, as well as this term chromatid, which I'll introduce in our next stage. So in our last stage then, we saw the process of mitosis, where you go from one cell, and it will be a stem cell, where it divides into two identical daughter cells, ready for specializing so that the tissue can grow or repair itself. Now, there is an important difference that you need to be aware of when we're describing the chromosome during mitosis. And it's a couple of terms that do throw people off quite a lot. And they are chromosome and chromatid. Of course, they sound very similar, and they're used quite specifically in certain circumstances. Now, you will remember from our first topic that a chromosome is a long chain of DNA that contains genes, and there are 23 pairs of these in a human cell. Now, they are normally linear, so they just have sort of one line, okay? But in order for the cell to divide, it's going to have to make a copy of it so that the future two cells both have a copy each. And so before mitosis, the chromosome is going to duplicate, and it will be held together in the middle hinge point, and it will look a little bit like a cross, okay, as you can see here. So when we draw chromosomes, we're almost always drawing them as crosses because that's when we see them as scientists, okay? We don't really see them in normal cell stage because they're not condensed, okay? They only condense when they are ready for mitosis, which is why we typically draw them as crosses. But you have to remember that the two crosses, the two lines in the cross, they're identical, okay? You've got two copies of the chromosome. And that's where the term chromatid is going to come in. So on the screen, you can see my cross here. Whenever we are referring to the entire cross here, we are referring to the chromosome, okay? Because it's just two copies of it are still attached. So in those first couple of stages of mitosis, we're talking about the chromosome condensing and migrating to the equator. Now, the chromosome is going to get pulled apart, okay? And before it's going to get pulled apart, we're going to use the term chromatid to describe the individual legs, if you like, of the chromosome. So here, I've got one chromatid, okay? And here, by which I mean all of this, yeah, I've got my second chromatid. Okay, so to make that a little bit easier, let me just color them in separately. So we've got, let's just say, I've got this one here. So imagine all this side of the cross is this chromatid. And let's do this here. This side of the chromosome is this chromatid. Okay, but the whole thing is a chromosome. And let me just reiterate that with your two colors there. Okay, so the whole thing is a chromosome, but the individual branches of it are chromatids. Okay, so the chromatids will be pulled apart, and then once they're by themselves, we're going to call them chromosomes again because there's no two sections. It's just one long chain of DNA. Okay, so it's going to be called a chromosome again. So really, it's just being really sort of pedantic, but make sure that you're talking about the chromosome as the cross, as it's moving towards the equator, and the chromatids are going to be pulled apart, okay, and then they'll become chromosomes themselves once they're in their individual poles, okay. Now the last term I'm going to describe, we've used it in the last section, and it was centromere, and the centromere is this little circle here, where the spindle fibers attach, and that's the centromere. Okay, so as you can see, we've got three words beginning with C. Inevitably, that does cause some confusion. Okay, so remind yourself that, maybe sketch this down yourself. Okay, make some flashcards with some terms. Okay, the chromosome is the complete set of DNA. The chromatids are the individual branches of a duplicated chromosome, and the centromere is the point where the spindle fibers attach for splitting the chromosome. So welcome back. We're now going to have a look at what stem cells are, what they do, and where we find them. Okay. Now stem cells are a very popular topic in biology. You would have covered them lower down the year because of their applications in therapy. Okay, so there's a potential wealth of treatment options we're using stem cells in the future because of their unique ability to divide and then differentiate or specialize into cells we might need for repairing damage in our bodies. So, by definition then, a stem cell is an undifferentiated cell. Undifferentiated means it hasn't specialized. That can divide to produce new cells. So let us do a quick summary of what the definitions are. So they are unspecialized, okay, and they have the potential to divide and differentiate into specialized cells. Try and spell correctly. Okay, so for example, our blood is made up of a variety of different cells, red blood cells, different types of white blood cells, platelets. And in order for you to replenish those cells, we need stem cells that can divide to produce new blood cells. But that stem cell itself is not a blood cell, but it will split, and the daughter cells will become either red blood cells or different types of white blood cell or a platelet, depending on how it's been, like how it's growing. And that happens in our bone marrow. Okay, and that's an example of tissue stem cells or adult stem cells, which are able to divide into a narrow range of cells. Okay, so we can find them in our tissues, ready to repair and to grow when we need to. But not all tissues. So, for example, if you've had a spinal cord injury and you lose your ability to control your limbs, that doesn't come back because for some reason we aren't able to grow new nervous tissue in our spinal cord. Okay, and that could be a potential avenue for treatments using stem cells if we can then put stem cells into a damaged spinal cord, for example. Okay, so the point is then, some tissues have them, some don't, okay? But they are commonly found in tissues, particularly those that are regularly needing repaired or growth, so skin, for example. Okay, so let's make a note of that. They can be found in tissues and differentiate into a narrow range of cells. Okay, but there is, of course, a very popular stem cell for the potential use of treatments that can divide into any type of cell. And of course, if you think about the development of an organism, at what stage do we have cells that can become any cells? Well, that's going to be right at the very start of the organism, okay? When it is an embryo. You have a fertilized cell, a fertilized egg cell. It divides into 2, 4, 8, 16, etc. And at that stage, all future cells, well, they come from those 16, don't they? So those cells can divide into anything, okay? And those embryonic stem cells are the kind of the golden boys, okay? These stem cells are the ones that we could potentially use for treatment of a variety of different diseases, okay? So we also need to say they are found in the embryo and differentiate into any cell, okay? So let us just recap that. So stem cells then, they have the potential to divide into and differentiate into specialized cells. They're unspecialized themselves. They don't have a particular job other than to divide. They're found in tissues and they can differentiate into a narrow range. So for example, in the bone marrow, you have stem cells that can divide into blood cells only, not skin cells, but a variety of blood cells. However, those that are found in our embryo, they will become our future organs. And so they can potentially divide into any type of cell and have a potential application in a variety of uses. Okay, so let's just recap then what the stem cell does. So you can see I've got my little baby stem cell here. Okay, it's going to be quite simple. What it can do is that it can either self-renew itself to produce new stem cells in that process of mitosis. Okay, let me just use my pen here. Mitosis. Okay, or it can become a specialized cell. So here I am going to use a different color here, so it can differentiate. Okay, and it can become then a specialized cell. So for example, it could become a white blood cell, or it could become a red blood cell, or it could become a platelet. Okay, it will differentiate into a special cell that has a particular job to do. Okay, it's that simple. There's only one more thing that we need to mention about stem cells, and that's stem cells in plants. They do the same sort of thing. Okay, they can differentiate into a range of cells and are needed for the plant to grow and to repair itself. The only difference really is the words that we use to describe them and where they're found. Okay, so whereas in an animal, stem cells will be found embedded in the tissues around the body, in plants they're only found in, well, two real places. Okay, and I'll do a sketch of a plant here. You will find that the stem cells are either going to be, so if this is growing here, here's our leaves, okay, some roots here. The stem cells are going to be found either in the absolute tip, okay, or at the end of the roots here, okay, and we call those meristems. Okay, the apical meristem here, don't worry about that term, okay, but we have the meristem here. You think about it, plant grows from the top, and the root meristems here, and again, roots, they grow out, don't they, to find their water. So they are the two sources of stem cells in a sort of a normal plant. Our final part of this revision video, and that is looking at the hierarchy of organization in multicellular organisms. Okay, you can see I've got some icons here on the left, and they represent the different stages of how we organize life that is multicellular and complex. So thinking about your plants, yourself, your pets, okay, anything that has multiple cells, anything that has organs, you know, that type of thing. Okay, so we're going to start with the basic unit. That basic unit, of course, is right down at the bottom here, and that is the cell. Okay, and the cell has a particular function to do, a specialized cell. Okay, so remember that our last bit there, the stem cells divide to produce new stem cells, or can then produce cells that become specialized cells. So, for example, you might have an individual muscle cell, for example, okay, that has special abilities to be able to contract. Now, that cell is going to be relatively useless by itself. So what you'll see is that cell will find itself amongst a cluster of identical cells in order to carry out that same job. And we call that collection of identical cells a tissue. Okay? And a tissue is made up of only that type of cell. So, for example, a muscle tissue is only made up of muscle cells. Okay? And together they'll be contracting, and because they're working together, you're going to actually start to see contraction and have the tissue contracting itself. So the cell itself isn't particularly useful by itself, but in a collection as a tissue, it starts to have some function. Okay, now different tissues can come together to form organs. Okay, so the organ here, for example, is the stomach, and the stomach is going to be made up of muscle tissue, okay, as well as things like connective tissue. And there'll be the inner lining of the muscle will be another type of tissue that can then secrete enzymes and acids. And so you can see that there are different tissues here that do different things, but they work together for an overarching job. So the stomach is to churn and to digest physically and chemically food. Okay, the muscle is going to contract to move the food along and to break it up by churning. Okay, the inner lining of the stomach, those cells there in that tissue will be releasing enzymes to help digest the food chemically. Okay, so you've got more than one tissue doing their own job but working together for an overarching functionality. The last section, this hierarchy, is the organ system. And the organ system is made up of multiple organs that do their own specialized job but work together for some much larger function for the organism to survive. So, for example, here we have the digestive system. It's got the stomach that is chemically and physically digesting food with its separate tissues. You then got, say, for example, the small intestine, a different organ that has different tissues that absorb those nutrients from the food that's digested. Okay? So, as you can see then, an organ system relies on multiple organs working together for a special job for the organism. The next level over that is, of course, the organism. So an organism is made up of multiple organ systems. Okay, so a human will have a digestive system, a circulatory system, a respiratory system, a nervous system, an endocrine system, a skeletal system, and they themselves are made up of all different organs that work together, which are made up of different tissues that carry out jobs that are made up of individual cells. Okay. So again, this is quite a common question. Perhaps I have the order. It might be a multiple choice question, and you've got to just choose which is the correct organization from smallest to largest, or largest to smallest. Be careful. Make sure you read the question carefully. Speaking of questions, we're now ready for the question part of our video. So see you very soon. Okay, so welcome back. Let's now have a look at a past paper question just to see how well we have revised today. It's very important when you're revising that you do practice questions after you've done a little bit of work. In fact, this is really where the revision really kicks in and supercharges. Okay, if you're just reading and watching videos, you're not applying yourself, so you don't really know how well you know something. Okay, so it's very important you spend at least half your time doing questions. Of course, there's only 3 marks here. 3 marks is not really enough for revision. This is just a way just to kind of just check to see how well you'll be doing. You should be doing, you know, at least for every hour of revision, you know, 20 minutes, 30 minutes of that should be looking at questions and marking it so you can check to see how well you have gone. Okay, but let's have a look at this anyway. So the first question here is looking at the stages of cell division. You've got to identify the 3rd stage by writing the number 3 next to it. Then you've got a picture of what's happening to the cell here. So why don't you pause the video, have a go at this question, and when you're ready, we'll come back and we'll look at the answers. Okay, so best of luck. Let's see how you've done. So, first question, the table describes some stages which occur during cell division, but not in the correct order. The first stage has been given. Identify the third stage by writing the number 3 beside its description. Okay, so let's have a look. You only need to do the third one, don't need to put them all in order. So let's start by looking at cytoplasm divides. Well, we know that happens at the end. Okay, so that can't be the third step. The nuclear membranes form, that again is at that last stage, just before the cytoplasm divides, so that can't be it. So it's going to be one of these two. The chromosomes move to the equator of the cell, or the pair of chromosomes are pulled apart. Okay, well, in order for them to be pulled apart, they have to be at the equator first. So the equator comes before being pulled apart, and these are the only two left, and we know the first step. So the number 3 has to be the pairs of chromosomes are being pulled apart. Okay, so that's for your mark. Next, the diagram represents a cell during one of the stages of mitosis. Name the part labelled L in the diagram. Okay, so this is the dotted line, it's growing from the pole here. This has to be the spindle fibre. Okay, nice and simple. Finally, during mitosis, a pair of chromatids was pulled apart, each moving away from the equator towards opposite poles at a rate of 1 micrometer per second. Calculate the distance between them after 20 seconds. Okay, so visualize it now. The chromatids are being pulled apart, okay, by the spindle fibers, and they're moving 1 micrometer per second from the equator. Okay, so after 20 seconds, it's going to be 20 micrometers here, and this one's going to be 20 micrometers here. So this is a bit of a trick question here. It's 20 micrometers away from the equator, but from the other chromatid, it's going to be that distance again. So the correct answer is 40 micrometers. So there you have it, cell division in a nutshell. Today we've looked at mitosis, as well as stem cells, and indeed how the hierarchy of organisation occurs in multicellular life. Apologies for Cinnamon's wee interruption there at the end of the video, though I'm sure you don't mind that at all, do we now? Until next time, guys, don't forget to like and subscribe, and we'll see you for our next episode on coordination and control. Take care now. Bye-bye.